Good morning, everyone, and welcome today to EAB University. Looks like we've got a good crowd coming in to hear our presentation today. My name is Robin Osborne, and along with my EAB University colleagues, Amy Stone from The Ohio State University, and Sarah Stack and Dr. Cliff Sadoff from Purdue University, we welcome you to today's presentation by Reed Johnson, who will be discussing the effect of EAB pesticides on bees and other pollinators. Now, Dr. Johnson is an assistant professor in the Department of Entomology at The Ohio State University's Ohio Agricultural Research and Development Center in Worcester. He received his bachelor's degree from Wabash College, his master's from Wake Forest University, and his PhD in entomology from the University of Illinois at Urbana-Champaign. His research focuses on determining how bees are exposed to pesticides and what this exposure has on the bee health and other pollinators as well. He has published more than 25 peer-reviewed papers on bees and bee health, and in addition, he teaches courses in pesticide science and beekeeping at Ohio State. Before we get started, I wanted to remind you that your comments and questions are welcome today. Please feel free to write them in the question and answer feature, which you can find by mousing over the bottom or occasionally the top of your screen. We will be making a note of all the questions, and Dr. Johnson will respond to them after the presentation so we can keep the webinar flowing smoothly. To keep these free webinars coming, we do need your feedback. Please stay tuned until the end when we will be providing a link to a survey that we hope you'll take the time to fill out. If you're one of the first 10 people to fill out the survey, we will be sending you an EAB goodie bag, but we hope you'll give us a feedback either way. This webinar is being recorded and it will be available for viewing soon at www.emerald-borer.info. You will also find the recordings for all previous EAB University webinars there. So I thank you for attending today, and
I just lost you, Robin. Can you still hear me? Can you hear me at all, Robin?
Yes, I can hear you, and we can see your screen.
Oh, excellent. Okay. Well, thanks for that introduction, and thanks to everyone who's listening and watching this morning, and who is watching this in the future in the recorded version. As Robin said, I work with honeybees, so that's my primary interest. So I guess I'm kind of on the other side of this. I know that the insecticide treatments used to control emerald ash borer are quite controversial among people working with bees because of the insecticides that are used to control emerald ash borer are excellent insecticides, and they kill insects, and honeybees are insects as well. So I think people are right to be concerned, but I'm going to just try to give you my take on the effects that emerald ash borer treatments are having on pollinators. So I guess to prevent you from thinking that you're going to get more out of this than you will, I will tell you the complete answer right now, and that is we don't know what the effect of emerald ash borer treatments are on pollinators because there just hasn't been a great deal of research done in this area. So what I'm going to tell you today is it's likely that emerald ash borer treatments could have anything from no effect all the way up to catastrophic effect. And as you can see, catastrophic effects of insecticides are not uncommon. Particularly in the past, here we've got an image of a whole apiary that was killed from pesticides applied to citrus. Now, I think we have good reason to suspect that emerald ash borer treatments are not causing a catastrophe because someone would have noticed by now if they were causing really severe effects on the survival of bee colonies in this country or in areas where emerald ash borer treatments are being used. It's more likely that they're having something down here in this lower range. It's entirely possible they actually have no effect on bees and that there's no problem at all. It's also possible there's some effect. It's just that it's an effect that's mild enough that beekeepers don't notice an overt effect on their colony health, which actually is quite reasonable. It's difficult to detect pesticide effects in bee colonies unless you are up to this catastrophic stage. I'm sure everyone's familiar with the long-term decline in managed honeybees in this country. Number of managed colonies peaked right after World War II with about 6 million colonies. We're down to about 2.6 million today. That number has been fairly stable for the last decade or so, possibly increasing slightly. Now, there's a lot of things that have driven this. Pesticides probably play only a minor role, if any, in the decrease in bee colonies. More of it's due to socioeconomic issues, people moving to the cities, and to the introduction of some pretty nasty diseases and parasites that are really hitting honeybees hard. But, I mean, being a beekeeper is a tough business, and beekeepers have suffered really what they consider to be unacceptable losses of colonies year over year for the last decade or so now. Beekeepers say that a level of loss of about 15%, which is in the gray bars on this graph here, would be acceptable. Beekeepers always lose some colonies. But what they've been experiencing is an annual loss of anywhere from 30 to 40%, and that's a real problem. Now, you may be asking, how can we be losing 30 to 40% of our bee colonies every year, yet the number of colonies in the country remains the same? Well, that's because beekeepers have a lot of management tricks up their sleeve, and they're constantly splitting colonies. So it's not like, a dairy cow or something where you have a set number of individuals, and if a cow dies, then you're minus one cow. With honeybee colonies, you can split them and use other management techniques to get your colony numbers back up, even though you may not have the total number of bees that beekeepers would have had in those colonies years before. So that's how beekeepers are squaring that circle, and it takes a lot of work. It takes a lot of input of money and effort to maintain and rebuild from these losses. So here's the real problem, is this knotty issue of all these bee health problems. In a previous version of this talk, given a couple years ago, I might have called this colony collapse disorder, where that term was ill-defined to begin with. So we're just going with bee health problems these days. And there's a lot of things. There's parasites and diseases, as I mentioned before. There's the drugs to treat those parasites and diseases. There's nutritional issues that are probably pretty severe in some cases where bees just can't find the food they need. And then pesticides do play a role, possibly interacting with the diseases and other things that are affecting bees. So it's this big knot of problems that is very, very difficult to solve. My primary interest in what role are these pesticides playing, and how are they interacting with these other factors to bring down the health of bees in this country. So before I get into the pesticide issues, I'm just going to give everyone a real quick background in bee biology and beekeeping here. So a colony that you see by the side of the road, standard white colony, it will have 3 different castes of bees in it. All honeybee colonies have a single queen, as probably everyone knows. Then they have anywhere from 3 to 60,000 worker honeybees, and those are, as the name would suggest, the bees that do all the work. These are the bees you see out foraging on flowers. And then, of course, there are the male bees, the drones, and depending on the time of year, there may be from 0 to hundreds of drones in a colony. You need all 3 of these castes for a honeybee population to be sustainable, because they not only need to exist, they also need to reproduce, and that's why you need to have the drones. Because these bees are not immortal; they do need to reproduce, just as all other biological creatures do. Often when we think of bees, we only think of the adults flying around, but there's all the developmental stages that insects go through are going on inside that colony. The queen lays an egg, and that egg stays in that state for 3 days, and then it will quote unquote hatch and undergo development as a larva, which takes about 5 days. Then eventually that larva gets capped over after it's full size and undergoes metamorphosis, so that 21 days after the egg was laid, you get a new young adult bee emerging from that cell that can go about and do work for the colony. And worker is the right term for these new young adult bees. They do an incredible number of tasks, and they've got it. It's really an amazing way they've got the division of labor arranged inside a bee colony, so that the younger bees do all of the indoor tasks like cell cleaning, cell capping, tending the brood, these nursing activities where they actually will consume nectar and pollen and secrete this jelly that they then feed to both the worker larvae and the queen larvae. You probably have heard of royal jelly. Workers get a similar version, but it's called worker jelly. So these nurse bees that are young adults spend a lot of their time eating pollen, eating nectar, so that they have the energy and the resources they need to secrete this stuff that is kind of like breast milk, honestly, that they end up feeding to larvae. And then, they do comb building, grooming, and as they age, they start to do tasks that are more associated with the outdoors. Food handling as the foragers come back, ventilating and guarding, and then finally, the most dangerous task of all is foraging. Only at the very end of a bee's life does it graduate to this truly dangerous task where it flies out of the colony to find food. And this is dangerous work. They might get picked off by a bird. They certainly wear themselves out, and they wear their wings down in the act of foraging. So this is a really elegant system. You put the bees that have the least life left in them, you make them do the most dangerous task. It works pretty well. Now, bees forage over an incredibly large range, and up to 3 kilometers is the standard. So you have this area of 2,800 hectares that the bees are foraging over for nectar and pollen, which is a tremendous area. And there's all sorts of, from a pesticide perspective, there's all sorts of interesting pesticides that may be used over this 2,800 hectares. Of course, they're foraging for the 4 things that bees collect, and it's pretty amazing that bees build this entire social organism and all of the comb out of just these 4 things. They bring in nectar and pollen. Of course, the nectar provides the carbohydrates for the colony. The pollen provides the protein and lipids. And then they also need to forage for water, depending on the time of year, to cool the hive or to liquefy honey, and propolis, which is this bee glue that they use to seal up cracks in the colony. And really interesting stuff. Now, a bee colony with up to 50,000 individuals is that's a lot of biological material in there, and they need a lot of food to sustain it. So a colony actually needs about 300 kilograms of nectar over the course of a year. That represents about 7.5 million foraging trips, and 20 kilograms of pollen, representing 1 million foraging trips. So this is the reason they need to forage over this 3-kilometer radius, is because they need a lot of food, and they need to travel over a large area to get that food. So that's a little background on bees for you, and I think it'll help you understand how what's used for emerald ash borer treatments intersects with pollinators, honeybees in this particular case. So I guess I'll take a look at this from a more formal risk assessment perspective. And if you look into the field of risk assessment, in the field of regulatory toxicology, you find that risk, which is the effects that we're talking about here, is the product of the hazard and the exposure. So in the case of emerald ash borer treatments, hazard is going to be the intrinsic insecticide toxicity of those treatments that are applied for emerald ash borer. The exposure is the bee collection of ash pollen and the insecticide concentration of that pollen that bees collect. Now, for there to be any risk to bees, you need both hazard and exposure. If you use an insecticide that has no toxicity to bees, the risk is going to be 0. Similarly, if you use an insecticide that has high toxicity to bees, but the bees never come into contact with it, they have no exposure, the risk will also be 0. So we're going to try to put these pieces together to really get a better assessment of the risk that bees are sustaining when emerald ash borer treatments are used. So we're going to start off by looking at the bee collection of ash pollen on the exposure side of the equation. So I kind of stumbled into this because we had a question in the lab about what bees are foraging on in agricultural landscapes during corn planting. And this work was related to the very clear insecticide exposure that honeybees receive during corn planting. And this is due to the insecticide seed treatment that's placed on most corn seed that's planted in this country. Now, corn planting happens late April through May here in Ohio. So we set up some apiaries around Ohio to look at what bees are foraging on during this corn planting. whether we see any dead bees during corn planting, and assess the pesticide levels in the food that bees are bringing back during corn planting. Now, this also happens to be the time of year that the ash tree is blooming, and we were a bit surprised to find that there actually is a lot of ash out here in this area of central Ohio, and the bees were foraging on it. So what bees are foraging on in an agricultural landscape is an interesting question, because if you look out there at in the spring at these very agriculturally intensive landscapes, you find that there is not a lot for bees to obviously be foraging on, because a lot of it's just crop field that is generally pretty clean, and there's not a lot of blooming anything in there. So we set up these apiaries out there in among the cornfields, and here you start to get a sense of where the bees really are foraging, because it's not all crop field out there. There are margins, there's roadsides, and there's residential areas. And in Ohio, we have these tree lines, which include ash trees. Now, just a little bit more about the amazing biology of the bee, and that's that honeybees, when they go out to forage for either nectar or pollen in this landscape or in any landscape, they display what's known as floral fidelity. And floral fidelity in bees has been known for millennia. This goes back to Aristotle, where he was reporting then that on each expedition, the bee does not fly from a flower of one kind to a flower of another, but flies from one violet, say, to another violet, and never meddles with another flower until it has got back to the hive. Now, the product of this floral fidelity is that when a bee goes out to forage for pollen, it only collects pollen from one particular plant type. So you have, bees that specialize on clover are only going to bring back clover, pollen, or nectar. It's not going to mix that up. And the product of this floral fidelity is that if you put a pollen trap on a colony, and there are these are commercially available pollen traps, the bees enter the colony, and they go through this series of screens that scrapes the pollen off of their legs, and you can collect it in this little tray here, this drawer that you pull out, and you get these pollen balls that are all interestingly color-coded. And each of these pollen balls is from one particular kind of plant, because each bee has 2 pollen balls that it can carry on its legs, and each pollen ball is specific. All that pollen in that ball came from one particular kind of plant. And so you can kind of use just color to start to determine what bees are foraging on. You can certainly see here, this is a time course of what bees are foraging on over time from mid-April through mid-May, and you can see that on a weekly basis it changes pretty drastically over a short period of time what bees are foraging on. This is all from the same hive as different flowers come into bloom and the bees' preferences change over time. So we took a more quantitative approach to identify the pollen sources that bees were visiting during this time period of late April to early May. To do this, we took a 10% subsample of this pollen that we collected from the pollen traps. And these pollen traps will collect potentially kilogram quantities of pollen. It's no kidding that these bees will collect a large amount of food over a relatively short period of time. So we took a subsample of that. We sorted it by color so that we would have relatively uniform groups of pollen here. We weighed each category of pollen to get an idea of how much of that type each color class represents, how much of the pollen came from each plant type. And then we put these different color fractions onto slides to do the definitive microscopic analysis. And so to do this, we just did a basic fuchsia stain, and you can see the basic fuchsia on these microscope slides here. And then we apply the pollen in a slurry to this basic fuchsia, and you put it under a microscope at 400x, and you can, along the bottom here, see the individual pollen grains. Now you can use a key to key out these pollen grains to figure out what kind of plant they came from, or the keys are very hard though, as most keys are. The easier way to do this is you actually just go collect some pollen from the plant itself, so you know what plant the pollen came from, and then you can match up that pollen from the reference collection with the pollen that the bees are bringing back, associate that with the color of the balls, and there you've got a pretty good sense of how attractive each plant type is and how much of the pollen came from each type of plant. And so here's, just to give you an idea of the spatial and temporal variation of the pollen sources over 3 sites that we looked at in 2013 in central Ohio. This is from April 29th through May 15th, really just a small section of time, but it's quite labor intensive to do this. What we found was that during this, late April, we found that foraging on a lot of herbaceous plants, the dandelions, mustards, and other herbaceous things that you'd expect bees to forage on. These are things that are growing in the roadside and in the fields out there. And here we've got them broken up by the different apiaries, apiary 1, 2, and 3 out here in the cornfields of central Ohio. Now, if you look down further, you'll see that there are rosaceous trees and shrubs that the bees are foraging on at this time as well as this ash pollen, which was in orange, and some maple in brown. Now, at the time that we saw this, we were surprised because we generally thought that bees did not forage much on ash pollen. We didn't think that they foraged much on these woody plants, with the exception of rosaceous trees at all. We thought that they would prefer the dandelions and mustards, which were quite abundant out there on the roadsides at this time. But there it is, the ash pollen, as we found by microscopy. Now, if you look over time, so here we have the hawthorn balloons down here that are other woody species. Now, corn planting happened from May 3rd to May 16th, and you can see that concomitant with that corn planting, the bees switched over to the rosaceous trees, and then they switched over to the willows and honeysuckles that began to appear later in that time. It's only early in this time period, and probably going back possibly into more mid-April, that the bees are using the ash extensively. And even extensively here, we're talking about, if you look on the average, it's anywhere from 10 to 20% of the pollen they're bringing back in this agricultural environment is ash pollen. We were still surprised to see it because bee biology is amazing. There, all through this period, there's abundant dandelions blooming along the roadsides, but for whatever reason, the bees are not preferring to go to those dandelions that are clearly highly abundant. They're going to ash trees, maple trees, and these rosaceous trees instead. Why that is, we don't know, but for whatever reason, they seem to prefer these woody things to the herbaceous things that we thought they would actually prefer. So I guess the take-home message from this, for us anyway, was that the herbaceous things that we see bees foraging on may not necessarily be what bees are really preferring. They do a lot of foraging in the woody environment, in these treed areas on the edge of the cornfields, but we don't really see bees foraging there because they're way up high, and it's very difficult to observe bees in that environment. It's much easier to see bees foraging on dandelion than it is to see bees foraging on a maple tree. And this is despite the fact that, most of this environment was crop field, but the bees were definitely preferentially using these semi-natural and residential areas where these woody species are growing. So, bees are collecting from these trees even though they have abundant access to alternate food sources in the form of all of these dandelions and the mints that are blooming this time of year. So I guess this is these trees are really underappreciated in their contribution to bee nutrition, and I think that kind of as a side, I think that they're really underappreciated, and that if we're looking to sustain pollinators in our landscapes, that we need to look at shrubs and trees as well, and they have a real important role to play in providing food for pollinators too, even though it's the herbaceous stuff that people seem to be most interested in. So we found ash pollen in our pollen traps, and we identified it using microscopy. We also identified it using a new method that involves high throughput DNA sequencing, a metabarcoding method. And I'm not going to belabor this, but we're just to go over real quickly how we helped work to identify what species are in this mixed batch of pollen using modern DNA sequencing techniques. And this has a lot of advantages because that microscopy pollen identification is very, very difficult and requires some serious expertise. So sequencing is a very attractive option if you want to know what's in a batch of bee pollen, but you're not an expert on pollen in that area, because you just take a subsample like we did before, homogenize it, extract the DNA using standard kits, amplify it using standard PCR techniques, and then you sequence it we use the Illumina MiSeq platform, We process the reads, do blast alignment with the sequences for the ITS2 region, which is what we used here, that are abundantly available in GenBank for all the plant species. We used this Megan 5 bioinformatics pipeline to do the analysis, and that gives us a plant taxa list. Now, we compared our metabarcoding results with our microscopic results, and at this point, we are really only confident to the level of family, and this is mostly because limited by our ability to identify pollen by microscopy anywhere below plant family. But you'll see that, so there's not complete concordance between these two methods, but the Olaceae, of which the ash tree is a member, was consistently identified in both the metabarcoding and the microscopy results. And if you look at the actual blast alignments, there is very good alignment between our reads and the reads that are in GenBank for this ITS2 metabarcoding region. So we're very, very confident that the pollen we collected is indeed ash pollen, and our bees were foraging on ash during this period. So we're fairly confident that bees may collect, up to 20% of their pollen from ash during this spring period. So the risk for exposure, the exposure is there. There is some risk, but the question next would be, what is the insecticide concentration in that ash pollen that the bees are collecting? Obviously, these trees are unmanaged in these tree lines, but if these were managed trees that were receiving treatment for emerald ash borer, what would be the insecticide concentration in the ash pollen? And I'll tell you right now, we don't know the answer to this question. We can just start to make some predictions. So systemic insecticides are used for the control of emerald ash borer. These include the neonicotinoids dinotefuran and imidacloprid through soil treatments. They're also used in trunk injections, both imidacloprid as well, also emamectin benzoate and azadirachtin through the trunk injection method. And then there are these systemic basal trunk sprays that make use of the neonicotinoid dinotefuran as well. And I think it's, just as an aside here, one of the real problems with the soil treatment method that is kind of regardless of the ash issue is that this soil applied treatments can potentially get into the nectar and pollen of other plants that are highly attractive to bees. This is just some clover growing a lawn here in Worcester, and if an ash tree was planted here and was treated with a soil treatment, and there wasn't a large enough zone that had been cleared of bee attractive plants, it's highly likely that imidacloprid in that soil drench is going to appear in the pollen and nectar produced by these other plants near to that ash tree. And I think it's very likely that it's going to be a whopping dose in these other little plants that are growing around here. And this is a real risk, I think, that with these soil applied treatments, is that it can get into other flowers that are attractive to bees, and it can pose a real problem through that route, irrespective of the systemicity inside the ash tree itself. So here's the list of all the different insecticide formulations and active ingredients that are potentially used to control emerald ash borer. And this is from the Insecticide Options for Protecting Ash Trees from Emerald Ash Borer by Herms, McCullough, Smitley, Sadoff, and Crenshaw. And the neonicotinoids play a pretty major role here. And I think that that's the reason that a lot of people get nervous about treatment for emerald ash borer, is that the neonicotinoids have a real bad name because they are highly toxic to bees, and people have a immediate reaction to them that if this neonicotinoid is being used and bees are exposed to it by collecting this ash pollen, that it could be harmful. So, but that all depends on the dose. What kind of concentration of insecticide is in the pollen that these ash trees are producing that have been treated with either through a drench or a trunk injection or through the systemic bark spray. Well, we can make some predictions based on the concentration of pollen of these neonicotinoids in pollen in other systems. So in corn, and I mentioned already that corn gets heavily treated with neonicotinoids as a seed treatment at a rate of 0.1 pounds per acre of active ingredient. I haven't seen this published anywhere, but I've seen a number of talks where they look at the level of pollen contamination in corn pollen, and it's always less than 10 parts per billion, which is a fairly low level. I'll put this number into context here in a minute. Other systems, now ash and tree applications are a little different than corn, obviously. For one, the rate is higher. For imidacloprid and dinotefuran, the annual acre use limit is 0.4 and 0.54, which is substantially higher than the 0.1 used in corn. Additionally, for trees, obviously the trees do not cover the entire acre, so this annual acre use limit is being applied to a relatively small portion of the acre in the form of these trees. So it's really just a fraction of the acre that's really being treated. But these ash trees are a lot bigger than corn plants, so it's just not really clear how well you can use what's showing up in corn pollen to predict what's going to show up in ash pollen. But at least it's a start. We can look at some other species as well. Canola. There was a nice paper in Nature where they looked at seed treatment of canola at the rate of 0.07 pounds per acre, and they found pollen concentrations at 14 parts per billion on average. Josephine Johnson, when she was at University of Maryland, she looked at the effects of imidacloprid on maple, where they used it, I'm assuming, at 0.4 pounds per acre, and she found it at 5 parts per billion in the maple pollen. So if it's hard to know, these systemic insecticides can be tricky in the way that they move inside a plant, and it's not necessarily, I mean, these other plants can be kind of a guide, but we can at least begin to form a range of the likely concentrations of neonicotinoids or insecticides in the ash pollen. It's likely to be anywhere from 5 to 110 parts per billion, just based on the use rate per acre in these other plants. I mean, it could be higher than this, it could be lower, I don't know, but I think this is a good range to start with. I will say at this point as well, we did collect a small amount of ash pollen from a few of the trees that have been treated here at the OARDC in Worcester that had been treated with imidacloprid the year before. And we submitted those for pesticide residue analysis, and they came back at 60 parts per billion, which is right in the middle of this range that we were expecting to see. So probably about 50 or 60 parts per billion is what you could expect in ash pollen treated the year before, at least based on our very limited results. So, okay, there is insecticide in the ash pollen. Bees do collect ash pollen. Now we really need to take a look at the hazard side of this equation. What is the toxicity of these insecticides to bees? Now you hear a lot about neonicotinoids being very toxic to bees. And how do you know what level of toxicity they pose to bees? Well, a good resource is to go look it up in the EPA's Ecotox database. And they have listings for all the different insecticides that have ever been registered. You can go look up the active ingredients in here and find their LD50s and the no observable effects levels on honeybees and a number of other species as well, if that interests you. So here I just looked up dinotefuran for honeybees, and you get this kind of semi-indecipherable chart. But over here, here we have the LD50, the oral LD50 of dinotefuran for honeybees, according to the EPA's database, is 0.0076 micrograms per bee. You can look this up for all of the treatments that are used for emerald ash borer. Dinotefuran, 7.6, I'm going to convert to nanograms per bee here. Imidacloprid is 3.9 nanograms per bee. Emamectin benzoate does not have an oral toxicity in it, but it has topical toxicity of 3.5 nanograms per bee. And then azadirachtin appears to be much less toxic to bees, though there aren't a lot of data in the database on this. The topical adult toxicity is 6.1 micrograms per bee, though the larval toxicity may be quite a bit higher than that. Really, I think more work is needed on azadirachtin before we have a sense for its toxicity. But I guess the message here is that, we have dinotefuran and imidacloprid here, which are neonicotinoids. We also have emamectin benzoate, which would be seen as an alternative to neonicotinoids, but in reality, it is not any less toxic to bees than the neonicotinoids are. So I would not call that a safe alternative to neonicotinoids. You might be better off just using the neonicotinoids where we have a little more knowledge about their effects on bees. I don't know. This is definitely an area where additional research is desperately needed to to know more about the toxicity of emamectin benzoate and azadirachtin to bees. So this I'm going to take a moment on. This is my slide rule that I use to determine where we are in the range of toxicity of a particular exposure to bees. So we've got and this is all based on that EPA LD50 data that I showed you, and based on the EPA's conversion chart that they can use to convert, how much pollen does a bee eat, how much nectar does a bee eat per day, and you can start to get a handle on how toxic a particular exposure is going to be for bees. So on the top here, we have the oral dose in nanograms per bee, everything from 1/100th of a nanogram all the way up to 7.4 nanograms. The line below that, we have pollen concentration, and this is in parts per billion, everywhere from 1 part per billion all the way up to 760 parts per billion. The range that we're predicting for ash pollen would be here in orange, anywhere from about 5 to 110 parts per billion. Though it may be higher or lower. This is kind of a guess. Below that, just for your reference, I have the nectar concentration from 1 up to 27 parts per billion. Now, you should note that it takes quite a bit of insecticide in pollen to equal a relatively low concentration in nectar. And this is just the result that bees eat a lot more nectar than they do pollen. So we really do want to be cautious about the amount of insecticide in nectar, because it can have a much greater effect at a lower concentration than insecticide in pollen. Now below here, I've tried to put the toxicities for imidacloprid, dinotefran, and emamectin benzoate in context here. I have the LD50 on the right-hand side of this chart for each of these insecticides. The LD50, and this should be the LD50 or LC50 in the oral exposure of around between 3 and 5 nanograms per bee for the oral dose. If you convert this into the concentration in pollen, this is around, 400 or so, 400 to 500 parts per billion in pollen. You would expect to see acute bee mortality in adults. I mean, that would correspond to a range of about 10 to 15 parts per billion in nectar. So if we just draw our lines down here from the pollen concentration, we would expect pollen to not reach the level of concern, which is, in EPA parlance, the level of concern is 40% of the LD50. Level of concern is the point at which mitigating measures are required to limit bee toxicity or limit bee exposure to a toxic insecticide. Years and years of using this have shown that if you can keep the levels below this level of concern here, which is 40% of the LD50, you can reduce bee losses to less than 10%, which is a good thing. This prevents any sort of catastrophic losses. But this is the point at which we begin to get into sublethal effects. So we don't really know much about the sublethal effects of dinotefran or emamectin benzoate. There have been extensive studies done on the sublethal effects of imidacloprid. And James Cresswell did a review paper a few years ago on the sublethal effects of imidacloprid, and he found that down to about 0.13 nanograms per bee in an oral dose, you see up to a 20% reduction in field performance of bees. So I'm using that as the end point for this sublethal range. And as you can see, the concentration that's in ash pollen falls well within the sublethal range. And that at the upper end of this, it's quite likely that the bees consuming this ash pollen could be experiencing some of these reductions in performance, or this kind of drunken stupor that bees get when they consume neonicotinoids. This remains to be seen, however. This is just my prediction here. I don't know of anyone that's actually tested to see if there are these sublethal effects when honeybees actually do consume ash, pollen from a treated ash tree, to see if there's actually any sort of sublethal effect. But it's certainly within the realm of possibility, and again, really requires further investigation. So does sublethal exposure to pesticides affect bees? And this is a somewhat controversial issue. I mean, there's not much disagreement over what a dead bee means. But at these concentrations, it's unlikely that you're ever going to see a dead bee. Instead, it's going to be this kind of range of sublethal effects, which in my experience generally manifests itself in these kind of drunken bees that are not capable of doing the work that you would expect of a worker bee. There have been a number of good experiments done on the behavior of foragers using, RFID tags and things like this, where you can actually see fairly profound changes in foraging behavior and foraging ability of bees when they're exposed to sublethal quantities of a neonicotinoid insecticide. The most famous study in this vein was done by Henry et al. It was published in Science back in 2012, and they found that if you take bees a kilometer away from their colony and release them after they've fed on a neonicotinoid insecticide, you start to lose bees because they just have this homing failure to make it back to their nest. And this is at a level of 1.3 nanograms of thiamethoxam, which is right there at the end of that range that I showed you on my slide rule chart. So there's clearly behavioral effects that are possible in this range. And then Henry et al., they put their results into a model, a computational model of a honeybee colony. And this isn't the model they used, but I like this image of a model here. And you can actually model the complicated dynamics that are going on inside a bee colony as eggs are developing into larvae, and larvae developing into pupae, and eventually become in-hive workers, then begin foraging. And then, these in-hive workers are also involved in brood care of the larvae. And it's a very complicated system. And if you start losing foragers, what they found is when you lose foragers, these in-hive workers that would have been caring for larvae suddenly get promoted to foraging because the colony needs to maintain their intake of food, even if it's at the cost of rearing some additional larvae. And over the course of time, you would expect to see a decrease in the population of a hive exposed to sublethal concentrations of a neonicotinoid, even though you never actually see a single dead bee. This colony just really fails to thrive in the way that they would have if they hadn't met that neonicotinoid challenge. So here's the real problem, I think, with the ash pollen, is that this ash pollen is out there collected by forager bees, and it's these young nurse bees that are tending brood that are consuming most of the pollen that's brought into the colony. These are relatively young bees. It's unlikely that they're going to die from this exposure, but that exposure to neonicotinoids in ash pollen may cause them to accelerate in their transition to foraging. Because it's pretty clear that when a young bee is not very good at nurse tending the brood for whatever reason, whether that's because it's poisoned or it has a disease, they quickly get promoted to foraging, because all bees go to foraging before they die. So these bees that are experiencing a exposure from ash may get promoted more quickly to foraging, which would decrease their lifespan and may have really major effects on the demographics of the bee colony, even though we're not actually seeing any dead bees from this level of exposure. So here's the summary of what may be happening here. So this chart that I'm showing you here is the adult bee population in a bee colony over time, and we have adult bees on the y-axis and time over the x-axis here. Coming out of winter, you have a relatively low bee population. The goal of a beekeeper and the bee colony is to build that population up over the spring so that they have a maximum population during this target period, the nectar flow, to collect as much nectar from the clover or the locust, black locust, or whatever that main flow is in your area, so that they can produce as much honey as they possibly can, so that the beekeeper can sell it and make money, so that the bees have food to survive the following winter. What pesticide exposure in the spring may be doing is reducing the ability of this colony to build up, so that you don't actually have any dead bees, but you have a smaller colony that is not as able to build up to a population where it can collect as much honey over the summer, and it has lower chances of surviving the winter, and certainly won't make as much honey for the beekeeper. So this is the scenario where you have no dead bees. It's just the bees that didn't exist that are the problem here, and that may be a real consequence of insecticide exposure in this early spring period. more research is needed in this area to figure out exactly what this impact is of early spring insecticide exposure. So just in summary, we've looked at the risk equation here. And at the beginning, I said we don't really know what the effect of emerald ash borer treatments is on pollinators, and that's still the case. I've spent almost an hour here talking about it, but we still really don't know what the effect is. We do know that there is some risk, though. We know that the insecticides used as emerald ash borer treatment have high bee toxicity, and they probably have sublethal toxicity at the rates that bees are likely being exposed to them. We do know that bees collect ash pollen, and we do know that the insecticides are likely to be present in ash pollen if it's used in the control of emerald ash borer. We're not talking about catastrophic losses here, but it's likely that there is some effect of this exposure. It's also likely that there might be no effect of this exposure. This is really where more work is needed to figure out where on this continuum these ash treatments fall, somewhere on the left side of this chart. But whether that effect that they may be causing is acceptable or not is going to be an issue of some debate, because what's acceptable to a beekeeper, what's acceptable to a homeowner or to someone else, if their ash tree dies from emerald ash borer, these are competing interests, and those are the kinds of knotty problems that will need to be worked out. So my recommendation, I think that the soil application are the riskiest, and I think that's because they have the chance of getting into the other kinds of flowers that are growing there at the base of the ash tree, and that may pose a much higher risk to bees if they come into contact with the pollen nectar from those little herbaceous plants growing under the ash. Dan Herm's recommendation has always been that you want to maximize the time between application and ash bloom, so that treatment has the maximum time to dissipate in the ash tree before the bees are back collecting pollen again. So that would mean applying these treatments just after ash bloom in the spring, so that you have almost a full year until the bees are back collecting ash pollen. Choice of insecticides, just because something's a neonicotinoid or not a neonicotinoid, in my opinion, is irrelevant. This emamectin benzoate is just as toxic to bees as the neonicotinoids that are also used. So there's no clear, compelling reason to switch to emamectin benzoate over the neonicotinoids. I think that decision should be based on which is better at controlling emerald ash borer in the particular situation. I don't think protection of bees really needs to enter into that, because they're all pretty much equally bad, at least at this point. So I'd like to I did not do all this work by myself. I want to especially thank Dan Herms, who made available his ash trees and has been really, really helpful in teaching me about control of emerald ash borer. I don't think I did his work justice here, but he's really taught me a lot about emerald ash borer situation. I want to thank members of my lab, specifically Rodney Richardson here in the back. He did all of the metabarcoding analysis, and Chia Lin, who did the hard work of the identification of pollen by microscopy, and all the other lab members for helping to maintain these colonies and collect all this pollen. So at this point, that's the end, and I'm happy to answer any questions.
Okay, we have a few for you. Carol Strayer had a couple of questions. The first one is, are there any differences in protein value for various pollen plant types?
Absolutely there are. The different pollen can have very different amino acid composition. Doug here actually is in the process of doing that for some of the pollen we've got, and he will be doing that for ash pollen as well, to figure out, to see what the differences are. Now, I should have mentioned that pollen has different nutritional value to bees, but bees are notoriously bad at choosing pollen that's good for them. They really don't have the tools to be able to say this is a really good pollen or this is a really bad pollen, because the only way they know what the nutritional content of that pollen would be after they consume it. Because the foragers that are out there collecting the pollen, they don't actually eat the stuff. They're just bringing it back to the colony. So they really have a difficult time assessing the quality of pollen. And I think that this is part of the reason they just collect everything they can find. And I think that that probably gets, so whatever nutritional deficiencies may be in a particular pollen source, it kind of gets averaged out by just collecting everything you can find. And that probably provides the bees with a balanced diet in the end.
Okay, the next question is, has there been any research on comparison of nectar for plant types, and where are bees getting their nectar?
So nectar varies even more than pollen does. Some nectars are very, very concentrated sugars. Some of them are much less so. And that bees do learn very quickly. Bees will go to the most concentrated sugar in the nectar that they can find in the landscape. They're very good at recruiting to that, and the whole waggle dance and the amazingness of the waggle dance and bee recruitment is all centered around getting the most sugar out of the environment that they possibly can. So there's a wide variability. Of course, ash trees don't produce pollen or don't produce nectar, so they're not attractive to bees in that respect. But I think that the nectar is the reason the bees really were starting to go to the rosaceous trees and shrubs, because those rosaceous trees and shrubs are generally produce a very concentrated nectar, and that's the reason that the bees like them.
Okay. Dr. Sadoff wanted to know, have you published this bee toxicity slide rule?
No, but I need to. I'm talking with the Pollinator Partnership. This was part of the work that they funded, and they would really like to produce it as kind of a. I'm about to work with a graphical designer to make a more user-friendly. This is my researcher product of slide rule, but I think it could be really useful to help people interpret the concentrations they're seeing, because the EPA has done all the hard work of how do you convert between these different matrices, and I think having a slide rule would be very helpful for a lot of people. So I do hope to get that published in the near future so that everyone can make use of it.
Cliff also wanted to know, can you comment on your thoughts about the hazards of emamectin benzoate given its 3-year longevity in an ash tree?
Well, the longevity is something I haven't really explored, but I think that if it continues to kill emerald ash borer larvae over 3 years, there's a good chance that a good concentration is present in the pollen over a similar period. So that definitely could be a mark against emamectin benzoate, is if it has a longer half-life in the tree.
All right, thank you. Do you have any guess whether azadirachtin will have sublethal effects? It seems that the dose lethal to bees is very, very high, and it may not.
It may not. There's been very, very little research done on azadirachtin in bees. So the answer is I just don't know. The EPA database has 4 entries, and that was the only LD50 I could find in there. It does appear that azadirachtin is more highly toxic to the larvae than it is to the adult bees. So there may be issues with that insecticide inside the hive for the larvae. But no one knows with azadirachtin. But it certainly would appear to be a pretty favorable alternative. There's a pretty major difference between it and the alternatives.
All right, thank you. When a tree is injected with the treatment, do you know how much will get back into the soil and be taken up by other plants, or is it only a danger for soil treatments? Mostly what I want to know is, can nearby flowers such as hosta flowers planted at the base of an ash tree take up any of the insecticide if it is injected into the tree?
I really don't know the answer to that question. My understanding is that these are systemic insecticides, and they're injected such that they should go up, but I may be wrong on that, and they actually can go down and can leach out of the roots kind of in a reverse systemic action into the soil. I don't know the answer to that question, but hopefully someone on here can answer that.
All righty. Another question, do bees collect conifer pollen, and is there any evidence that bees would collect hemlock pollen?
I do know that they collect conifer pollen. You can find it in moderate quantities in some of the other samples that we've done. I think that the assumption should be that if a plant produces pollen, there's a high likelihood that bees collect it. I think their strategy is just to collect everything that's pollen-like. They will collect. Whether you can see bees doing it or not, it's likely that. That's why it's amazing to see these bees are really a good sampler of the entire landscape. They bring back everything blooming, ends up back in the hive.
All right. It would seem that there's a difference in exposure of rural bees versus urban bees from ash treatments. Treatments of ash trees is only likely in residential settings, so the source of exposure to pesticide will be limited to urban bees. What are sources of exposure of imidacloprids likely in rural settings?
Well, imidacloprid is used as a seed treatment in on soybeans. So in a rural setting, that's probably the principal method of exposure, at least here in Ohio. Yeah, those ash trees out there on those tree lines are, I mean, their days are numbered. No one's treating them, I'm sure. So yeah, it would be very interesting to do a similar thing in an urban setting where there are ash trees being treated, just to see what component of the bee's diet this time of year is made up of these ash trees, and to see what, you know, in an urban setting where this is more relevant, what is the risk to the bees there?
All righty. Dr. Sadov did comment that you are correct that insecticide does go up and not down as far as when it's injected. So, okay, we have another question here. The person says, great presentation and loves the slide rule. I would comment that exposure to neonics from clover, etc., after soil application would be reduced by mowing, which is some of Dan Potter's work. Further, in using neonics, one does not want herbaceous plant material growing at the base of the tree. Anecdotally, we have seen lowered benefits to the ash tree if there is an extensive root sharing of the soil injection zone by shrubs and perennials. So prepare the site, clear out the hedera, daylilies, etc. And then I think if there was reverse flow, we could kill periodical cicadias. Just guessing.
Yeah, well, I think that just underscores that. I mean, there's a number of good reasons to keep other things out if you're doing a soil treatment. Now, I don't know, the mowing situation, I think you would probably want to be pretty diligent with the mowing to keep the, you know, other things from blooming around the base of the tree. But it's probably best for the treatment just to remove everything as best you can, so that there's no competition for that insecticide with the ash tree.
Okay, I am going to bring up the survey slide here. Hold on just a second. I'll get that done. And go from there. But this has been great information. I've learned a lot about bees from this.
Well, good.
A lot more than I had ever known about bees. So that's great. Folks, if you could please fill out the survey and get that to us too. Are there any other questions for Reed while we are at it here? Let's see if I can. I was hoping. Are you seeing the survey slide? That's what I'm kind of worried about right now.
I'm not seeing it.
Okay, then I'm not doing something right here. Okay. Maybe I need
there we
to
go.
do it this way. Okay, so this is what I've got to do. There we go. There's our slide. If we have other questions, make sure that you, okay, we think we might have one here. Okay. Cliff just wanted to tell you did a great job.
Thanks.
Okay. I think from, at least from here, we're good. Is there, what I wanted to know is we have been having, we had a couple of questions of people asking if you would share your information as far as like a, you know, your slides, you know, make a copy of the slides for, or a PDF or, you know, just a handout of the slides. I didn't know if you, I told them that they would have to contact you for that. That's something that we don't deal with. So, let's see, if you can, let's see, I'll put in the Q&A here what your email is. And it's 5005 at OSU, Johnson.5005 at OSU.edu. Is that correct?
That's correct.
Okay. All right, I've got that on here. Okay, I have one other question for you here. Did you choose not to measure the insecticide concentrations in the pollen or just resource limitation?
So we did measure the insecticide concentrations in the pollen. But we were interested in the amounts related to corn planting, and we definitely did find insecticides in the pollen collected during corn planting. But I think it's irrelevant for the ash because none of those ash trees were being treated, I'm assuming. We certainly didn't find detection before planting began when the bees were really working that ash pollen. It was only after the corn planting began that we really began to see any level of clothianid and thiamethoxam in the pollen bees were bringing back, which are not the neonicotinoids used in ash anyway. Those are the corn seed treatments. So it doesn't appear that there was any level of contamination of the ash pollen out in those rural environments. All right. Well, thanks, Robin.